Good afternoon. I'm Dr. Brian Shapiro, a marketing segment manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled From Curiosity to Breakthroughs: Accelerate Your Drug Development with Assay-Ready Cells, presented by Drs. Fatah Kashanchi and Heather Branscombe. Before we get started, I'd like to say a couple words about ATCC. Founded in 1925, ATCC is a century-old nonprofit organization with headquarters in Manassas, Virginia, and an R&D and Services Center in Gaithersburg, Maryland. We are the world's largest, most diverse biological materials and information resource for cell culture. We are the gold standard. We're also an innovative R&D company featuring new product formats that support drug development, safety testing, and high-throughput screening, such as the assay-ready cells that we will present today. In today's presentation, I'll give a brief overview of ThawReady assay-ready cells and introduce our speakers. Dr. Branscombe, our first speaker, will highlight the challenges in generating working cell banks in post-thaw recovering of cryopreserved vials. She'll then go on to present a solution that allows you to go from frozen to data in one workday without the need for additional cell culture or expansion. Drs. Branscombe and Kashanchi will then show the utility of these assay-ready cells in multiple applications. Dr. Branscombe will share the results she found in extracellular function assays using ThawReady as the cell model, and Dr. Kashanchi will then show the utility of these assay-ready cells in two widely different applications, using ThawReady to examine HIV infection and then to examine downstream signaling of drugs of abuse. To get into the overall problem, as we all know, cell-based assays have lengthy timelines. It's generally due to the requirement of cell expansion, essentially all the processes needed to get to a synchronized cell stock. You start with cell banking, which can take months of passaging, cryopreservation, and expansion to go from your seed stock to your master cell bank and working cell banks. Then, in phase 2, getting to your data, you must thaw, recover your cells, and then plate them before you run your assay and finally see your data. The process is lengthy, it's risky in that it opens yourself up to misidentification, contamination, and genetic drift. It's costly in regards to human capital and reagents. The ideal solution for this bottleneck are assay-ready cell systems that can be used at thaw, showing similar functionality as the parental cell line that you use in your assays. And so that's what we developed. ThawReady, a new line of ready-to-use cell products. We have ATCC ThawReady assay-ready cells, which allow you to go from frozen to data in just one day. You can simply plate your cells directly and start running your assays immediately. Similarly, we have ATCC ThawReady 3D spheroid kits, which are pre-plated spheroids that provide a uniform spheroid in every well. Well, with that, I'd like to introduce our speakers. Our first speaker is Dr. Heather Branscombe. Dr. Branscombe is a senior scientist with ATCC. Throughout her 17-year career, she has gained broad experience working in both academic and industry settings. She has extensive experience in cell and molecular biology and completed her graduate training in biosciences from George Mason University. While at ATCC, she's held positions in manufacturing, quality control, and technology transfer to support the production and qualification of cell lines and other critical biological reagents to support the scientific community. In her current role, she manages a team of biologists to support the CDC's International Reagents Resources Program, as well as other government contracts. Since 2018, she has played a key role in establishing and maintaining ATCC's extracellular vesicle portfolio. In this role, she was responsible for developing and validating large-scale EV manufacturing protocols and performing various EV biochemical and functional assays. Her current research is focused on advanced methods for EV purification, characterization of novel EV subtypes, and mechanistic studies of stem cell-derived EVs in different models of cellular repair. Our second speaker is Fatah Kashanchi, a tenured professor at George Mason University. Dr. Kashanchi received his PhD in 1990 under the supervision of Dr. Charles Wood, who also worked with the Nobel laureate Dr. Susumi Tonegawa at MIT. He then moved to National Cancer Institute at NIH's intramural program and continued his work on RNA viral infections with the late John Brady on HIV and HTLV transcription and chromatin complexes. He is currently a tenured faculty in the Department of Systems Biology at the Prince William campus of George Mason University. He has obtained independent funding of more than $28.9 million since his departure from NIH in 2000. He's also published more than 270 peer-reviewed manuscripts and served as an editorial board and reviewer for a number of journals, including Cell, Molecular Cell, Nature, Nature Medicine, Science Translational Medicine, Retrovirology, etc. He is a regular NIH study section member and has served on 163 panels and chaired 21 since 2000. By the way, if you have any questions for your speakers today, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. Any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website at www.atcc.org. So with that, let's welcome our first speaker, Heather Branscombe.
Thank you, Brian, for the introduction. Today I'm going to be speaking about some of the application data that we've generated as internal and external beta testers for the ThawReady cells. A brief overview of the topics we're going to cover. Again, we're going to touch on the challenges with bioassays. We'll give a brief overview of the ThawReady system, and then again, we'll highlight some of the key application data that includes post-thaw recovery, functionality testing using EVs, which are extracellular vesicles, and lastly, HIV-1 infection and drugs of abuse. So bioassays are very essential tools that are utilized to advance our knowledge of biological processes and to evaluate the biological activity of different substances. However, traditional bioassay platforms do come with several challenges, and I think it's important to address these. These challenges include cost and resource dependencies that could ultimately significantly require investments in materials and labor, and also complexity and variability that's inherently associated with the use of biological systems. In addition, there's always an increased risk of contamination when you scale up cells prior to initiating a bioassay. And lastly, and perhaps most importantly, there's a post-thaw recovery period that is required to allow cells to recover from cryopreservation. And depending on your cell line, this can take anywhere from a few days to a couple weeks. So ATCC's solution to these challenges was the development of our ThawReady cells, which are a standardized consumable cell product that can be directly used in different endpoint assays. Essentially, this allows for a thaw, plate, and go type of format. ThawReady cells yield high reproducibility and high functionality, and most importantly, they've been formulated by our cryobiology department with a very unique cryogenic storage media, which eliminates that need for a post-thaw cell culture. So for these reasons, ATCC ThawReady cells really have a tremendous potential to greatly streamline and standardize bioassay workflows. ThawReady cells are derived from THP-1, which is one of ATCC's top-selling cell lines. THP-1 originated from the peripheral blood of a patient with acute monocytic leukemia. And these cells are extremely versatile and widely used in the biomedical research community because they serve as a model system for studying monocytes, which are critical for studying the immune response, inflammation, and as well as studies relating to infectious diseases. And in particular, they have really high value for immuno-oncology research, which is vital for developing cancer immunotherapies, as well as for toxicology research, because they can be used to assess the safety and the efficacy of new drugs. ThawReady cells are, again, they're an assay-ready luciferase reporter cell line that's been specifically engineered to express the firefly luciferase gene, LUC2, which is under the control of an NFkB promoter. So by design, ThawReady cells are able to monitor the activity of NFkB signaling, and this is critical, again, for regulating the immune response, inflammation, as well as cell survival. And the cartoon on the right is just kind of illustrating this concept. It shows activation of NFkB, which ultimately is going to drive the expression of the reporter gene, and this is going to produce a detectable signal. In our case, this is luminescence, and the intensity of the signal directly correlates with the level of NFkB activation. So this gives us a nice way to measure the pathway's activity in a quantitative manner. Collectively, these features enable ThawReady cells to be utilized across multiple different applications, such as drug screening and development of anti-inflammatory therapies, pathogen detection, as well as basic and functional mechanistic studies that are relating to NFkB signaling and various biological processes. So I had the opportunity to serve as an internal beta tester for the ThawReady cells, and I'll be sharing some of the specific preliminary data that I generated in the next few slides. Just a disclaimer, my research involves extracellular vesicles, so I believe this is a somewhat novel and innovative demonstration of the THP-1 ThawReady cells in a novel application, both for the ThawReady cells and as well for the EVs. I'm going to give a brief overview of EVs. If you're more interested, I would direct you back to one of our earlier webinars that can be accessed on the ATCC webpage. But in the interest of time, I'll just highlight quickly what are the key features of these vesicles. So EVs are nano-sized particles, 30 to 1,000 nanometers in diameter that are released by all cell types, both under physiological and pathological conditions. They carry various components of the cytoplasm and the cell membrane, and they mediate intracellular communication. And collectively, for these reasons, EVs are utilized mainly as either diagnostic markers or therapeutic tools. And with respect to the therapeutics, it's really the stem cell EVs that have the highest potential for that field of research. Stem cell EVs are being considered a potential alternative to traditional stem cell-based therapy. They're more potent, they have increased stability, and they have a lower immunogenicity, which makes them very attractive targets, for the future of regenerative medicine. They also contribute to critical processes such as tissue repair and regeneration, and this is largely due to their anti-inflammatory and their anti-apoptotic properties. So for these reasons, stem cell EVs in particular are very important tools for studying cellular signaling pathways, which is why I rationalized that they would be a good tool to use in the ThawReady system. I've also included here, if you're interested in learning more about our preliminary data with stem cell EVs, we have 3 recent publications. So I've included QR codes here that should link to those, but we've already demonstrated that EVs have multiple different functions in various different cell types in vitro. So again, this was my justification for wanting to test them out in the ThawReady system. The first assay that we performed in our lab was to assess the post-thaw recovery of thaw-ready cells. So in our lab, we used the ViCell Blue analyzer, and we measured viability and cell yield immediately post-thaw. And this data shows us that the thaw-ready cells consistently had high viability. We had an average of about 90% across multiple different vials tested at different time points and different operators. And we had cell counts that averaged around 1 million cells per mil, which is equated to about 10 billion viable cells per vial, which is a yield that's significantly higher than what you would expect from a traditional cell line. And this yield allows us to immediately plate the thaw-ready cells for our bioassays. So to first assess functionality, we performed an LPS challenge assay. Again, the cells were seeded immediately post-thaw at a seeding density of about 50,000 viable cells per well. We treated with LPS from E. coli for a stimulation period of 3 hours, and we tested 3 different doses, low, medium, and high. And we used a bright glow assay platform with a readout on the biotech citation. And overall, we found from this initial study that thaw-ready cells were responsive to all 3 doses of LPS, which were tested. So this was good. It demonstrated that we could use this LPS challenge in a downstream or subsequent EV repair assay. So we next designed an assay similar to the one that I just described, where we treated cells immediately, excuse me, immediately post-thaw. They were seeded at the same seeding density, treated with LPS for a same stimulation period. We chose the low concentration of LPS to test for this study, and then we added different types of EVs. Namely EVs that came from stem cells or EVs that came from a non-stem cell. In this case, we were doing cancer cell EVs, and we also included a positive control budesonide, which is an anti-inflammatory drug. So this was a time course study where we seeded multiple plates in red at different time points. On day 1, we performed our first bright glow assay. And as you can see from this data, our background cells, this is our control, lane 1, had no detectable luminescence, which was expected. Lane 2 is our LPS treated cells, which had a robust response even after 24 hours post-stimulation. And lane 3 is cells that were stimulated with LPS and received a single treatment of MSC EVs. We do see that MSC EVs, as well as HeLa EVs, which again are cancer EVs, after 24 hours were lowering the relative expression of the luminescence. And this was also further demonstrated by the positive controlled drug budesonide in lane 5. So this tells us that 24 hours post-stimulation, post-treatment, we are seeing anti-inflammatory effects from all of the different treatment conditions. Our next readout was on day 2, so 48 hours after stimulation with no additional treatments. Here we see that there's still a robust readout of luminescence in lane 2, which is LPS treated. And interestingly, in lane 3, we see that stem cell EVs continue to lower the level of NFkB activation as compared to HeLa EVs in lane 4, which the effect appears to have worn off. This is also seen in lane 5 with the positive controlled drug, where the level of luminescence is similar to the MSC EV treatments. So this is showing us that over time, there is a difference in the different treatment conditions. And this was most drastic after day 5, where we're still detecting in lane 2 a signal from LPS. So there is still an activation here that's being able to be detected after 5 days. But in lane 3, we see that HTRT MSC EVs have continued to lower the NFkB activation, again, in a statistically significant manner. And we do not see this in the case of cells that were treated with HeLa cancer EVs. And also in lane 5, we see that the effects of the positive control drug have worn off. So collectively, this data shows us that stem cell EVs are able to exert a pretty potent anti-inflammatory effect over time. And this is not the case with our positive control drugs. So the EVs, it suggests that they have a pretty significant half-life that's able to persist in culture over time, and also shows that our non-stem cell EVs, in this case cancer EVs, are not having a good effect, which again, if you look at day one, you would suspect that maybe cancer EVs and MSC EVs both have anti-inflammatory effects, but over time, you see that that effect wears off, and you see the strongest response in those cells that were treated with MSC EVs. So we're really excited by this preliminary data. We are planning additional experiments using ThawReady cells to continue to evaluate the anti-inflammatory properties of stem cell EVs in response to other inflammatory stimuli as well. And lastly, before I turn it over to Dr. Kashanchi, I did include this data. It was the same assay setup as I just previously described, except our readout was a cell titer glow assay, which is going to correlate to cell viability. So this is not a luminescence luciferase assay, but we just wanted to make sure that our cells were not undergoing any adverse or cytotoxic effects. So on day one, we performed cell titer glow. We saw that the average RLU of all cells were similar to the control, which is very good. And by day 6, we did another assay again, just to make sure that over time, we weren't seeing any adverse or negative effects of any of the treatment conditions. And there were no statistically significant differences. So this just gave us confidence that our results in our luciferase assay were not being skewed by any other factors that could be inducing cytotoxic effects. And with that, I will turn it over to Dr. Kashanchi to go over his application data.
Thank you very much, Dr. Branscombe. It's a pleasure to work with you guys. We've been working together for the past several years, and we're very excited to use these cells, especially for viral infections and also drug screening. So again, thank you for this collaboration. Next slide, please. So what I want to tell you today is a little bit about the utility of these cells. And again, I'm sure many of you are familiar with HIV infection. HIV really comes in many different shapes and forms and flavors, so to speak. And the one that's pretty predominant in North America is clade B. And to the left, bottom left, you see all of these different clades. And they certainly have very different genomes, these different clades. And also there's clear epidemiology that it's infecting different populations throughout the world, and they behave quite differently. And of course, you can tell that by when using antiretroviral treatment, drug treatments. So what we wanted to do is to ask a very simple question and start the discussion with the clade B of HIV and ask, if HIV enters these monocytes, will we see any NFkB activation? And the reason NFkB is a big deal for us, the reason is because HIV has different levels of NFkB activation. For instance, clade C has a very different, the one that's predominantly in India and in South Africa. That clade has quite a robust activation of the NFkB pathway, whereas clade B is somewhat borderline in terms of its activity when it comes to NFkB induction. So we wanted to start with the clade B and figure out, and that's the data I'm going to show you today. The clade B that we chose is called 89.6, and it is a dual tropic virus. It infects both T cells and myeloids. And of course, the literature on HIV is quite robust, and on T cells, and there's literature, I would say, maybe about 70% on T cells and 30% on myeloids. So we're very interested on the myeloids because they do end up going to tissues. They settle in tissues, including lymphatic and the lymph nodes, and of course the CNS. So we're very much into CNS type of abnormalities, especially when it comes to these viral infections. So next slide, please. So what I'm trying to show you here is the life cycle of HIV. Again, this is, I'm sure many people are familiar with this, but just to orient everyone, if you look at the left side around 11 o'clock, this is where the virus would bind to a receptor and a co-receptor, and they would enter the cell. Once it enters the cytoplasm, it goes through reverse transcriptase, and the RNA becomes a double-stranded DNA, and that incorporates into the genome in the nucleus. So, and then, of course, afterwards, if you have induction of gene expression, you would have mRNA synthesis, and eventually proteins are made, and the proteins are packaged into virions, and they would exit. Now, what's interesting about this pathway is that at the very early stages of infection, when a virus is touching the receptor and the co-receptor, signals, intracellular signals that lead to NFkB activation gets turned on. Whether NFkB activation leads into an interferon response, gene expression in the nucleus, or whether it leads into cytokine activation in the nucleus, that all is very possible. But at least the very first thing that happens is you have signal transduction turning on right after when the virus touches the cell. And at that point, you get eventually the NFkB induction. So we were very interested in that, and we wanted to see if the virus touches, basically at the time of touching the cell surface, will it be able to activate the NFkB? So these cells, these THP cells, were ideal for us. And by the way, one of the things we had to do is HIV enters inefficiently into monocytes, so we had to make sure we push these cells into a macrophage-like phenotype with an inducer, and that way we would have the receptor and co-receptor expressed so the virus could potentially enter. So next slide, please. So the experimental design is to take cells that are frozen. We immediately thaw them, and we use about 50,000 viable cells. We count, and then we use roughly about 50,000 vials, 50,000 cells per assay. We would treat them with PMA. So PMA induction is important because this is where you take monocyte into a macrophage-like phenotype. Then we would infect with HIV, and this is a dual tropic form. It's 89.6. That would infect both T cells and the myeloids. And we did a little bit of a titration, 100, 300, and 500 nanogram of P24. Just for fun, we threw in antiretroviral. Again, antiretrovirals that we've used are T inhibitors and protease inhibitors. They're not integrase inhibitors. Certainly, there's no transcription inhibitor in there, so it serves more of a control. And then the TNF is just to show in our hands that the NFkB is induced in these cells, even after you have the PMA induction. And we threw in a bunch of drugs, and I'm just going to show you one slide on this CBD and HU308, and we wanted to see if any of these would induce NFkB. Next slide, please. And the first thing that I wanted to show you is just a triplicate, biological triplicate of this very same experiment. If you look at assay 1 on the left side, the cells alone gave us a little bit of a readout. Of course, that's mainly because of the PMA induction. Then if we infect these cells with HIV at 100, 300, and 500 nanogram of P24, so we've equated the amount of P24 to the amount of viruses, you could gradually see that there's very nice induction of NFkB, and that's all the way to lane 4, and lane 5 serves as the positive control TNF at 10 nanogram per mil. Again, we repeated the same thing. That's assay 2 and assay 3. Very nice reproducible results, very clean. So again, this is serving as control for us for many of our assays. And of course, down the road, we're doing a lot of drug screening. And again, this is very useful for these drug screenings, as well as doing things like chromatin immunoprecipitations. But that's a different set of experiments. But the reason I bring this up is because the utility of these cells is quite enormous, and we're able to use them quite readily. Next slide, please. So if the virus touches the cell surface and it activates the NF-κB, that's fine. But then again, of course, the luciferase is turned on and you can see the expression of luciferase, but that's not sufficient for us. We wanted to make sure that the virus did get inside the cell and the RNA did go through a cDNA synthesis and eventually integrated into the chromosome. So that's the question we asked in panel A. 3 independent experiments, again, cells without any infection. There is no viral copy numbers in the nucleus. So this is a PCR, straight PCR for HIV genome. And then if you add 100, 300, and 500 nanogram, you would start seeing amplification of the HIV genome. And then, of course, if you throw in antiretroviral drugs, these drugs are against the protease, and yeah, of course, you're not going to be able to see a lot of transcription inhibition or even integration issues. So therefore, you have the genome still intact in the chromosomes. Panel B shows you a translation. So if the material is there, if the genome is there, will it still give you transcription and translation? And lanes 1 and 2 are the controls, and lane 3, of course, is molecular weight. So lane 4, 5, 6, as well as 7, 8, 9 are the parental cells that are not luciferase positive. So first we wanted to ask, if we do an infection in these cells, the parent cells for for our experiments, will we see viral entry? Will we see transcription and translation? And the answer is yes. So lane 7, 8, 9 is when you have the virus, and lo and behold, GP120, which is the envelope, is expressed. P24 is part of the gag, and that's expressed. And then the real experiment is lanes 10 through 15. This is where we use the luciferase cells, and lane 10, 11, and 12 have no viral infection. And 13, 14, 15, we do have viral infection in there. And again, these are the actual cells that have the luciferase open reading frame. And lo and behold, GP120 is expressed and P24 is expressed as well. So this basically shows you not only the virus is able to infect the parental cells, but also more importantly, it's able to infect the cells that have luciferase. So not much has happened with the receptor co-receptor right after addition of the luciferase into these cells. Therefore, you do see a nice viral entry and transcription and certainly translation. And panel C shows you just a fun experiment we've tried to do with CBD. It's not supposed to give you an NF-κB induction, but we have an analog of CBD, which we think it turns on the NF-κB, and this is called HU308. It's an analog of CBD, and it binds specifically to CB2 receptor, and we think it activates the NF-κB. And this form of NF-κB may not be important for cell death, but more for anti-apoptotic gene expression. So at this point, we're trying to design experiments to do chromatin immunoprecipitations right after drug treatment and to see which form of NF-κB is binding to the promoters. We're looking for specifically a P50, P65 heterodimers and also P50 homodimers, and trying to figure out which form is being activated and which form is binding to the promoters. Again, they require a little bit more cells, but very straightforward set of questions. We're just looking for the dynamics of NF-κB binding to promoters. Next slide, please. So what we've shown you so far, which I think is quite significant, at least from what you saw from Dr. Branscom's conversation, her part of the talk, LPS is clearly able to activate this promoter, and that's fairly standard. TLR4 is the target. And I think that's, the standard of the industry, of course. We've shown that TNF-alpha is able to do this as well in these cells, which is, again, another positive control. And in our case, when we're looking at viral infections, and of course, down the road, we're going to be doing drug discovery and use these cells for drug screening, we can tell that once the virus touches the cells, it's able to activate the NF-κB. This is important for us because many times we want to come up with inhibitors that would stop the binding of the virus to the cell surface and not even allow the virus to enter. So I think this particular assay system is quite novel for us. Of course, all of these three different pathways are very different, but the signal transductions, including MyD88 from LPS, MEK from TNF, and the AKT from the viral infection, they all lead into inactivating the IK beta in the NF-κB complex sitting in the cytoplasm. Of course, you have eventually the ubiquitination of the IK beta and degradation of it, and now you have this NF-κB complex, which, depending on how this stuff is made and how it will bind to a gene, to a promoter, it could be a P50 dimer or a P50 P65 complex as well. So we are very excited about these cells. We think they have a lot of utility, and it's really making life much easier for us. And so we're really super excited to continue working with ATCC on these particular experiments. And with that in mind, I'm going to turn it over to Dr. Branscombe.
All right, thank you. This is a quick summary of what we've discussed today. We've demonstrated that ATCC ThawReady cells are able to overcome many of the challenges that are associated with traditional bioassays. ThawReady cells offer high viability, high cell yields, and very high functionality in a very reproducible manner. And we've demonstrated some unique applications for using ThawReady cells. So we've shown that these cells can be used in assays relating to cellular damage. This can be in the case of damage with cancer, EVs, or viral infection, and also used in cellular repair assays. And in our case, we showed preliminary data using stem cell EVs, but this also extends, to other compounds that can be used for drug screening assays. And lastly, which Dr. Kashanshi was just mentioning, which I think is really a very versatile feature of these cells, is that they can be used to study multiple different mechanisms of NF-κB activation. So it allows a more widespread use of the cells. And we showed specific examples of TLR4, TNFR, and viral attachment to demonstrate this. So thank you for your time. And we would just like to take a brief moment to acknowledge all of our collaborators from ATCC as well as GMU who supported this work. And with that, we will turn it back over, and please let us know if you have any questions.
Well, thank you, Dr. Branscombe, and also thank you, Dr. Kashanshi, for the excellent presentation. In just a few moments, we'll begin our Q&A session. Please remember to use the chat function available through the webinar program to submit your questions. By the way, this session will also be archived on the ATCC website at www.atcc.org. So let's go ahead and jump in with our first question. And these first two actually look like great ones for Dr. Kashanshi. First of all, are there any differences in NF-κB activation in cycling versus non-cycling cells?
Yes, so thank you very much for the question. Great question. So in our experiments, what we've done is we've used PMA on THP-1 cells, and of course, PMA is important for differentiation into macrophages. So this is where you go from a cycling monocyte into a differentiated non-cycling macrophage. Now, we haven't done quite titration of the amount of PMA or some other inducers of differentiation, but what I can tell you is that to do that, first, I think we have to do a better titration of PMA and look for macrophage markers, including things like CD14, CD36, CD11B, and then, of course, follow-up for both M1 and M2 type of phenotypes for these differentiated cells. Now, will the NFkB levels be different between cycling and differentiated cells? The answer is yes. We clearly see a difference within cell cycle for NFkB. So we see a clear difference between, for instance, G1 cells versus G2 cells. And we do see differences, and I think we've published this a while back. There's a difference in terms of what binds to the HIV promoter. But be that as it may, so yes, there is NFkB differences within cycling cells. But more importantly, if the cells are differentiated, there's higher levels of NFkB to begin with. So that's probably why you do see this background level of the luciferase showing up after PMA induction. So the answer is yes. You're going to see differences between cycling and non-cycling NFkB levels, and that is going to come down to the promoter of interest, the complexes that are going to load up on these promoters of interest. But yes, there is a difference, absolutely.
Nice, nice. Now, Dr. Kishanchi, here's a question that's somewhat related. Do all RNA viruses cause NFkB activation?
So that's a great question. So the answer is yes, but it really is context dependent and titer dependent. So for instance, in the case of these THPs that have been stimulated with PMA, here we are banking on induction of receptor co-receptor for this virus to enter. Now, that means that if it does touch the surface, which we believe it does, and when it enters, because we do see RT activity, we see integration of DNA, then there is going to be NFkB induction without trouble. Now, if other RNA viruses require the same receptors, which they don't, COVID, for instance, SARS-CoV-2, some of the other also retroviruses, whether they're human or animal retroviruses, if the receptor is there, the chances are, yes, you're going to get high level of NFkB induction. If the receptor is low or not there, the chances are you won't have receptor binding to the virus. Therefore, you may not have as much NFkB induction. So I think the NFkB induction really correlates with what is on the surface of that cell in terms of the receptor and whether that receptor is functional for the virus to bind to. And if it does, the chances are, yes, there will be NFkB induction.
All right, great. Very complete answer. Okay, the next couple of them that have come in look like good ones to send Dr. Branscom's way. So, first of all, how does the cryopreserved format of ThawReady cells enhance their immediate usability post-thaw?
Hi, Brian. Thank you for that question. It's a really important and fundamental question, I think, when you consider how these cells were specifically engineered. And in order to answer that question, I think it's important also to understand what we mean when we say cryopreservation injury, because the process of freezing and thawing can actually have pretty significant damages to cells if the freeze media is not optimized and if your freeze profile also is too harsh. So there's a few different mechanisms that can contribute to this. One of them is intracellular ice formation, during which cells are cooled too quickly, and this leads to ice crystals that form within the cytoplasm. And the ice crystals can actually puncture the cell membrane, and this can damage intracellular structures, which can overall, again, disrupt the normal biological and biochemical processes that the cell would normally undergo. Another mechanism is osmotic intolerance. This is when the cells are unable to withstand osmotic stresses that are caused by changes in the solute concentrations during the process of cryopreservation. This can also irreversibly damage the cell membrane, which can ultimately lead to a membrane lysis and cell death. And lastly, one other point I want to touch on is the potential for the cryoprotectant, which is usually, you think of cryoprotectant, you need that to prevent some of this injury, but also if the concentration of your cryoprotectant is too high, under certain circumstances, this can actually cause osmotic imbalance. This can impact the metabolism of the cells, disrupt that, and this can also impact the integrity of the membrane. It can be compromised. And overall, this will have a net negative impact and will make the cells much more susceptible to cell damage and death. So ATCC has a specialized group, our cryobiology team at ATCC, who have meticulously optimized the cryopreservation medium as well as the controlled rate freezing profile to minimize those cryopreservation injuries that I just described. So the cells by nature were developed so that we would have an enhanced post-thaw recovery, which would enable the cells to immediately be plugged into bioassay workflows after thawing.
Nice, nice. All right. So here's another great question for you, Heather. What are the potential synergistic effects of combining mesenchymal stem cell extracellular vesicles with other therapeutic compounds in treating inflammatory conditions?
Well, that's another great question. I'll do my best to give a high-level summary, but this is an entire field of literature that's evolving. But MSC-EVs can be engineered by different methods to enhance their anti-inflammatory and their reparative properties. Some of these different mechanisms include you can do active loading, which you would use this technique to directly incorporate a therapeutic compound into the lumen of the EVs. And two examples of this method are electroporation or sonication, which basically the principle relies on the disruption, the temporary disruption of the EV membrane and then incubation with your therapeutic cargo. The membrane will reform and then the drugs are incorporated into the EVs. Another example is through the genetic engineering of the parental cells to express the therapeutic molecule of interest. This can be a nucleic acid or a protein. So if you engineer your parental cells to inherently express this, then the EVs that they naturally secrete will also have that desired cargo. And one other method is through surface modification. And this is something that we're actually working on right now at ATCC in a project. So you can use different techniques. One is click chemistry. So you can take a therapeutic protein or peptide and you can attach it directly to the surface of the EVs. The beauty of this is that it can enhance the efficiency of tissue targeting and drug delivery to different tissue types. So overall, I guess back to the question, how can this synergistically be used to treat different inflammatory conditions? If you combine EVs, MSC EVs with these compounds, you have the potential to enhance their ability to polarize macrophages, so going from that M1 inflammatory to the M2 anti-inflammatory phenotype, which will, again, have a really potent effect. And then you can also combine MSC-EVs through those methods that I just described with other anti-inflammatory drugs like corticosteroids or other broad-spectrum anti-inflammatory agents, and this can improve their ability to modulate and reduce the expression of various pro-inflammatory cytokines. So I think that overall, this approach of combining therapeutic compounds with EVs can lead to improved drug delivery by increasing the stability and the half-life of the drugs, and again, also enhancing their targeting ability and uptake in specific cell types.
Gotcha.
I wonder if I can add something to this. Is that okay?
Oh, absolutely.
Yeah. So I wanted to say that we have tested this very same question in our lab the difference between cancer EVs, using them as a vehicle for delivery, versus these MSC EVs. And we've been fortunate, we've used various types of these reparative MSC EVs. So we've used regular MSC EV, we've used H-TERT MSC EVs, and also iPSC EVs, versus 3 other cancer EVs. And these are PC3, there was HeLa, and I think there was also maybe a 293T, which has T antigen. What I can tell you is that the cancer EVs, they all equally do well in terms of repair in our hands. When it comes to doing extension of an experiment, especially an animal model, all of these cancer EVs cause trouble. And so I have stopped completely using cancer EVs, and that includes 293 EVs, by the way. We've stopped completely using these EVs as a means of delivery. However, the MSC and the iPSC EVs, especially the ones from ATCC that we've seen, there's quality control. There's actually, they know the content, they know the RNA in there, they know the protein, et cetera, the kinases, so on and so forth. Those EVs work like magic. They are fantastic for repair purposes. And then on top of that, we don't quite understand why, but is it protein, is it RNA, is it membranes, is it all of the above? So I don't think we understand the mechanism, but they are fantastic for repair purposes. And if you want to put stuff in there, things including drugs that are hydrophobic or half hydrophobic, half hydrophilic, including things like CBD, or if you want to put in microRNAs in there or small therapeutic RNAs, whether it's outside on the surface of the EVs or inside, they work, again, fantastic. So I'm quite impressed with that field. It's true what you heard from Dr. Branscombe. This is an emerging field, but we completely stopped for the past 2 years all of our cancer EVs for therapeutic purposes. Of course, we use them for diagnostic purposes. That's a different story. But for therapeutics, we've completely shifted to MSC EVs. And I'll stop right there.
Great. No, that's a very exciting angle, actually, to this work that you guys are doing. Okay. The next couple of questions that have come in are around the cell viability assays that Heather, you use. So the first one was, what concentration of bright glow and cell titer glow did you use to check your results in the stem cell EV experiment?
Okay, thank you for that question. When we perform both the bright glow and the cell titer glow, when we set up the assays, we make sure that the volume of our cells as well as the volume of our treatments comes out to a final volume of 100 microliters. And so following each of the protocols, these were Promega assays, Promega bright glow and Promega cell titer. So it's always a 1-to-1 ratio. So in this case, we used 100 microliters of each respective reagent and the assay, again, just to match the final volume in each of the wells.
Okay, that makes sense. And then for the second question, first of all, thank you very much for taking the time to present to us on ThawReady cells. The question is in regard to the cell titer glow after thawing the THP-1 ThawReady cells. Did you use the regular seeding procedure, or did you have any kind of changes, alterations to that?
Okay, thank you for that question. I believe we had a slide earlier in the talk that gave a high-level overview of our strategy. So when we did the cell titer glow and bright glow, we followed the same strategy for seeding. So immediately post-thaw, we count our cells, and then from there we seed 50,000 cells per well, again, depending on the assay, but the cell concentration is always the same for consistency.
All right, great. Now, the next question that's come in, I can go ahead and handle, actually. So do we have ThawReady prokaryotic cells? The short answer to that is we don't have ThawReady specifically, but we do have a new line of prokaryotic cells called microquant. And what microquant allows you to do is store the specific microbes at a higher temperature than usual. It's either at room temperature or it's at -20, and then you simply add the reconstitution buffer to the cells, and they instantly reconstitute. And by instantly, I mean under 60 seconds. And I've seen this demonstrated actually in front of my eyes. It's very impressive. So that product line is called microquant, and I strongly suggest you check it out. All right. And then we have a final question that's come in, so we'll make this last call. Please, if you have any final questions, submit them now. And this one looks like a good one for Dr. Kashanchi. So Dr. Kashanchi, could you talk about how extracellular vesicles help in HIV infection identification with P24?
So I don't know if I understand the question, but let me go back and explain what the experimental design was in our case. What we did was we incubated the virus with the cells that had been PMA treated. And then the virus enters the cell. There is clear NFkB induction. So you see the luciferase activity. The virus enters the cell. It goes through reverse transcriptase. It goes through cDNA synthesis, and then it incorporates into the genome. Once it's in the genome, there will be transcription by the RNA pol II of the cell. So you have mRNA, that mRNA makes it into the cytoplasm, and you translate that into a bunch of polyproteins, including things like P24 for the virus. So this is the cycle that we've used in our experiments. Now, how does EVs help repair HIV-infected cell? Well, that's a paper we published 2 years ago, and it's the second part of a scientific report paper. And there, what we found was that what we call good EVs, and these are the MSC EVs, these good EVs are able to reverse viral infection by lowering the amount of cytokines. And the 3-4 cytokines that we put in the paper, at least we could detect, were things like IL-8 and I think IL-6 was also in that mix. So the good EVs can repair damaged cells. We don't quite understand the complete mechanism, but we believe that it inhibits the viral spread. So what we're doing now is we're approaching everything from not only from the viral point of view, but also from the immunology point of view. The viral point of view, we may have drugs against the virus, or in some cases we don't. Actually, in most cases, we don't have drugs against many of the viral infections. And then the second thing is, okay, if you lower the amount of the full genomic viral material, what about all of the stuff that's left over from the virus? These are the so-called PAMPs, and the leftover material from damaged cells that are called DAMPs. What do you do with all that stuff? Well, the immune system has to clean things out, and that takes a lot of time, energy, ATP effort, so on and so forth. But we think that these good MSCVs, in fact, reverse that inflammatory response, and you get a much healthier cell that recovers much better. So that's our approach at this point. The good EVs are helping to dampen this overly hyperactive immune system.
Okay. Can I flip that question on its head a little bit? I think I figured out what the asker is trying to get at. So P24 is part of the detection system, however you want to say it, for fourth generation HIV tests. Does P24 antigen appear on extracellular vesicles coming from cells that have been infected by HIV? So it's kind of like, I know I've heard you repeatedly talk about how virus body parts wind up in some of these vesicles. Is P24 like antigen one of those?
So the answer is yes. We have a paper we published, another paper we published, and the title of, I don't remember exactly the title of it, but in a nutshell, we're trying to ask who comes out first. Do EVs come out first? And these are, by the way, EVs that have viral body parts in them. Do EVs come out first or the virus comes out first? And it turns out that right after induction of transcription in these cells, the EVs that come out contain body parts of the virus, but they are not virus, right? So they're missing 10, 15 different components. Now, if you wait long enough, in other words, if you go 24 hours or 18 to 24 hours, now you see a combination of virus plus a bunch of other EVs. And that particular virus in the 18 or 24 hours is infectious. But the stuff that comes out at 3 hours, 6 hours, and 9 hours, those are not viruses. They have body parts, including P24 in them, and they also have non-coding RNAs from the virus, including things like TAR. So yes, you can have EVs coming out of the infected cells, and those EVs, we've considered them as bad EVs, and we use them for diagnostic purposes. And yes, they could cause a lot of problem for the neighboring cells, for the recipient neighboring cells. And now how do we reverse that? Well, we don't know. We actually don't know how to remove these bad EVs from the system. But they do show up early on before the virus shows up. And of course, they will have kind of proteins in them, including things like P24. So some of the EVs may have high levels of TAR non-coding RNA. Some of the other EVs may have P24 in them. Some may have RT in them. So it's a heterogeneous population of EVs that are being shoved outside of the cell because the cell no longer can chop these things into pieces. Autophagy doesn't work. So has no choice but to ship these things outside. So that's what we've seen so far.
Nice. Thank you for bringing that full circle. All right. Well, I'd like to thank our presenters again for the excellent presentation, and thank you everyone for attending. Had lots of great questions come in. For more information about ThawReady cells, you can navigate to www.atcc.org/ThawReadyCells or use the QR code on the screen. Also, we have a podcast with the scientists who led the effort to create ThawReady cells. So you can listen to our podcast, episode 26, ThawReady, Transforming Cryopreservation for Instant Cell Functionality at www.atcc.org/behindthebiology. By the way, Drs. Branscombe and Kishanchi also appeared in our first season of Behind the Biology if you want to listen to them talk more about extracellular vesicle biology. So thanks again, everyone, for attending. Have a great day.